Hi everyone, this is Lonnie, the 4th year general pathology resident. I was asked if I could cover the basic transfusion testing of Type Screen and Crossmatch, so I hope this video helps you out. First, let's talk about determining the patient's blood type. What we do is we take the patient's blood sample and we spin it down and separate out the red cells from the plasma. We put the red cells into several little test tubes, and to each of these tubes we're going to add something different. In one, we're going to add anti-A antibodies, in another, anti-B, and in another, anti-RHD. That's going to give us our RH positive or negative. So when we take these antibodies and we add them to the tube, what matters is whether or not they find their antigen on the surface of the red blood cells. So for instance, this is inside test tube A. There that we're adding anti-A to. If the person has A antigens on their red blood cells and the antibodies that we add, the anti-A, find those targets, we let them incubate so they come together, they're going to bind. Then, just like we talked about for the Coombs test or the direct antiglobulin test, we add anti-human antibodies. So these are anti-human globulin. They recognize those globulins. And when they start to bind, it ends up linking the cells together because some of these can bind more than one at once. And the cells agglutinate. So if this person has A on their red blood cells, you're going to get agglutination in the anti-A tube. Just like that. So that's a positive test that shows this person has A on their red blood cells. If this person also had the RH antigen, they're positive for that. The same thing's going to happen when we add the antibody against that. We're going to end up with cross-linking and agglutination of the red cells. So this is what the blood type should look like for somebody who is A positive. For someone who was B positive, it would be exactly the opposite. The red cell tube wouldn't agglutinate because there's no A antigen for those antibodies to meet. But the B tube would because there's B antigen, so the anti-B antibodies bind. So that's what it should look like for somebody who is B positive. What if somebody was AB positive? Well, as you guessed it, now every tube the antibodies are going to meet their antigens on those red cells. So all three of them are going to be agglutinated. Finally, let's look at a Rh negative blood type. In fact, let's look at O negative. If somebody is O negative, then there are no antigens whatsoever on the surface of these cells for those major blood group antigens. When the antibodies come in, they have nothing to bind. They come and go, oh, there's nothing there. Gee, we can't even bind anything. So the red cells do not agglutinate, and all three of them would stay suspended. So that's what it should look like for a patient who is O negative. There was no matches between the antibodies we added and the antigens on the surface of the red blood cells. This is what we call the forward type. So to summarize, forward type uses the patient's red blood cells and added antibodies. Got it? Now let's talk about the reverse type. This time, we take the patient's plasma that we separated off at the beginning, and we add that to our tubes. We are looking in this test, just like the indirect antiglobulin test, for what antibodies already exist inside this patient's plasma. We're going to add donor red cells to the tubes that have a known antigen on their surface. So to this tube, we're going to add A cells. This one we're going to give B, and this one Rh positive cells. For a patient who has anti-B antibodies in their blood already, in their plasma. In the tube that we added the B positive cells to, the patient's plasma antibodies are going to end up binding to those cells in that tube. Then when we add our anti-human globulin to the tube, just like before, the cells are going to cross-link. So in this patient, we're going to get agglutination of those red cells down at the bottom of the tube. But the person doesn't have anti-A in their serum, so the A positive blood cells are going to just stay suspended, all kind of nice. And the person doesn't have anti-RH, so those are going to stay nice and suspended too. So can you figure out what blood type that is? This is somebody who is A positive, because their serum did not react with the A red blood cells, but it does react with B, because someone who's A positive naturally has anti-B floating around in their plasma. But they're not going to have anti-RH because they express the RH on their cells, so that one is not going to agglutinate. Let's look at one more example to make sure that this is clear. So let's look at the example of somebody who is AB negative. Well, in this case, the person expresses A and B on their blood cells. So in their plasma, they are not going to have either anti-A or anti-B antibodies. But since the red cells are negative for the RH antigen, all they have is A and B. Oh, sorry. This is the person's red blood cells, but we're going to add in the donor cells that are positive for the RH antigen. The person's plasma does have anti-RH in it, so those are going to bind, and then our anti-human globulin is going to bind and start cross-linking the cells. So what are those test tubes going to look like? Well, when we add A cells, there's no A antibody, so those are going to stay suspended. When we add the B donor cells, there's no B antibody, they stay suspended. But when we add the RH positive cells, this person has anti-RH antibodies in their serum because they're negative, so that's not something that naturally occurs. So that tube is going to agglutinate. So this is what it should look like for somebody who is AB negative in the reverse type. It's kind of the opposite of the forward type. It's a confirmation. So we look at what antigens are on the person's red cells and what antibodies are in the person's serum, and they should line up and match. It's like a double check. That's how we can confirm with great confidence that that is the person's blood type, and it helps us know what type of red blood cells we can transfuse them with. I'd encourage you to work through the other blood types, draw out the test tubes, see what they should look like for both the forward and the reverse type. Now let's talk about the screen. The antibody screen is also similar to the indirect immunoglobulin test that we did, or antiglobulin test. So for a screen, what we want to do is find out whether the patient has any other antibodies present in their serum that could react with donor red blood cells. So we take the patient's serum, put it in a whole series of test tubes, and then we add donor red blood cells that have a known antigen on their surface. There's all kinds of red blood cell antigens other than the major blood groups. There's Kell, there's Duffy, there's little symbols for all different sorts. When we add the donor red cells into the tube, if the patient's serum does not have that antibody, it stays suspended. If it does have the antibody, then they're going to agglutinate at the bottom. It's exactly the same as the other test we just did, because the antibodies will meet the antigen on the cells. This one I did Duffy. So the patient's plasma antibodies are going to bind, and then when we add our anti-human globulin, just like before, it's going to start cross-linking the cells together. So that way, looking at our whole panel, we can say which antibodies the patient has in their serum. So we are screening for antibodies that might react if we give somebody donor cells that have one of these smaller antigens on them. Now let's talk about a cross-match. The traditional way to do a cross-match is to take a small sample from a unit of blood that you think is compatible with your patient's blood. So here's a bag of A positive red blood cells. And there's a little label on the side and a barcode that contains information for the computer about what other antigens might be present on these cells out of those little ones. Maybe there's some Kel, maybe there's some Duffy. We know it's A positive and our patient's A positive, so that's a good start. But we need to know if there's any of those little antigens that might interact with what we screened for with the screen in the patient's antigens. So there's a little tiny side bag on top of a red blood cell unit that you can stick a needle in and draw out some of the sample. This is really important because then you don't have to stick a needle in the main bag and you don't risk introducing bacteria and other things that could make the patient very sick when you transfuse them. Then we take a sample from the patient's plasma, just like we did for the screening test, and we add the red blood cells from this unit directly into the tube. If the suspension stays all nice and homogeneous and suspended, we can say, oh good, there's no antibodies in this patient's plasma that are going to react with these red blood cells. This patient and this unit of blood are compatible, and we are going to take this unit of blood and set it aside for this patient. So you only order a cross-match when your patient is going to need to be transfused within the next 24 hours, because this blood unit is taken off the shelf, put aside, it's not available to anybody else, it's taken out of the pool. If, however, we tried to test this unit and the result was agglutination, again, after we add our anti-human globulin to bring it all together, then we would say, oh dear, there's an antibody in this patient's plasma against some sort of antigen on these red blood cells. And we might not know what it is because maybe it didn't show up in our panel of tests. Maybe it's an unusual smaller antigen, but if it's not compatible, then we will not be giving this blood to this person if there's agglutination. The other way to do a cross-match is electronically. So what I mean by that is if we have the results of our patient's screen test and they were all negative, so we can say this person does not have any antibodies in their serum or plasma that we know of. And this unit of blood, when we tested it on the barcode and the label, it doesn't have any weird, unusual antigens on its surface either. Then we can say, you know what, based on the fact that this screen was totally negative and that the computer information about this unit says it should be compatible, well, then we can trust that these two things are compatible. This is called an electronic cross-match. We don't actually mix the patient's plasma and the unit of blood to be given. We don't need to do that step because we have enough information to say that it's going to be safe. Only if the screen was positive, meaning the patient did show that there's at least some antibodies in their serum to some antigens, or perhaps if the person has had multiple transfusions, that's when we have to do the manual cross-match that I showed you in the first step. So I hope this has been somewhat helpful. I know it's a bit of a complicated topic, but if you work through a few problems, then you'll be able to sort it out. You can reach me anytime with questions at this email address. And all I can say is good luck on your exam.